So I would like to introduce our next speaker, William Stephenson from New York Genome Center, and he is doing a lot of cool stuff. And one of the things he's implementing and development we will try to reproduce tomorrow. I hope we will succeed, but today you will hear a talk about single-cell sequencing and open microfluidics devices. Please welcome.
Great. Can you hear me? All right. First off, I want to thank the organizers for inviting me to tell you about some of my work and some of the work of others in the lab that I'm in. My name is William Stephenson, and I'm a senior research engineer in the Technology Innovation Lab at the New York Genome Center. The title of my talk is Single-Cell Multi-Omics Using Open Microfluidic Instrumentation. Okay, so first, I just want to tell you a little bit about the Genome Center. We're a little bit new. We're located here near Soho in Lower Manhattan, and the Genome Center is essentially an independent nonprofit academic research institution. We're comprised of institutional members from many of the major New York City region hospitals, research hubs, academic institutions, and which you can see up here and down here. The New York Genome Center maintains research and collaboration agreements with a lot of these institutions while also providing vital high-throughput sequencing services. Okay, so this will be a technology talk. So I'll first start by talking about MiniDrops, which is an instrument that I developed to do droplet microfluidics, and it's low cost and 3D printed. That'll comprise most of the talk, but then next I'll talk about a technique developed by one of my colleagues called Dropseek, and this is sort of an extension to single-cell RNA-seq experiments where you can obtain surface proteomic information from single cells at the same time as you're performing single-cell RNA-seq. And then I'll talk finally briefly about cell hashing, which is an extension of Dropseek, which allows you to perform sample multiplexing and also identify doublets in your droplet microfluidic single-cell RNA-seq experiments. Okay, so this is kind of like the obligatory single-cell RNA sequencing technology slide. And so what we're seeing at the top are various technologies through the years that have been developed and extended to perform single-cell RNA sequencing. You can see very early in 2009, you were doing manual approaches in single tubes, gradually working our way up to plates, followed by microfluidic techniques, and then finally in situ barcoding. So this is a nice plot showing the number of single cells in each one of these studies as a function of time, different years here. And you see a general trend, and this is a logarithmic graph, right? So you can see a general trend here of increasing throughput, or the increasing number of single cells that we're allowed to study in these types of experiments. So this is really telling about the technology and how fast it's moving. In particular, I want to talk about droplet microfluidics, which has really played a pivotal role in expanding the throughput. Right around 2015, Klein and McCosco both came out with complementary papers that really kind of pushed the boundaries of the number of cells we can study with these techniques. So these techniques, they're called Dropseek and InDrop, and for the most of this talk, I'll be talking about Dropseek. They're very complementary and similar. Essentially, you start with a complex tissue. You can isolate the cells into a single-cell suspension. And then these cells are actually co-encapsulated into aqueous droplets surrounded by an oil phase using a microfluidic chip, such as shown here. Now, these beads, these microparticles are unique in that they're coated with oligos that are bar-coded, unique to each microparticle. However, the oligos on these microparticles are also uniquely indexed, so that you know, after downstream preparation of these libraries that you're performing in these droplets, you can actually collapse PCR duplicates. So essentially what happens is you encapsulate a bead which captures the mRNA from a cell that's lysed because the bead, the microparticle, is in lysis buffer. So we can make lots and lots of droplets very fast, and this allows us to get very high numbers of cells. And then we can kind of perform some of these, you know, RT and other reactions in bulk, finally leading to a library that can be sequenced. InDrop is very similar, however, the beads are a gel type bead, so you can actually load them more reliably into droplets more effectively, increasing the number of cells that you can study. So microfluidic instrumentation is typically either developed as a bespoke instrument, sort of shown here in this picture. I'll refer to this as like a research grade instrument, or typically you can also buy microfluidic instruments, right, in commercial forms. The costs are very different, pretty high for both, depending on what types of experiments you have going on in this research equipment. Generally, a research level setup is going to have high flexibility, very low portability, and moderate ease of use. The flexibility is due to all these components, namely syringe pumps and microscope attachments here, which can be reconfigured for any type of particular experiment one's interested in performing. Now, commercial instruments are typically very expensive, not very flexible because they're often configured for one purpose and to perform that one purpose very well. But they can be more portable, and they're typically very easy to use because they've come from a company that's, you know, developed this instrument to be very robust. Now, I thought there might be room for an instrument that maybe falls in between these two on the spectrum. And this is what I came up with. So it's called MiniDrops, and it's open source. It's relatively inexpensive, much less than $1,000. Typically, when we build it, it's around $600. I seem to think it falls somewhere in between both of these setups in terms of flexibility, because you can do multiple droplet microfluidic experiments on this instrument, and also highly portable and moderately easy to use. So this is a breakdown of the instrument. It's essentially a multi-tiered instrument, and this is the top view, and then you can see the different levels of the instrument going down. So the black pieces here are actually 3D printed. You can see the variety of components here. There's a touch screen on the front. There's a custom printed circuit board which houses the necessary electronics and pneumatics to operate the instrument, along with a camera here. So the instrument is controlled through a Raspberry Pi single board computer. Essentially, this kind of takes input from the user at the touch screen to perform experiments that are going on. So also, I've used a Raspberry Pi camera in conjunction with a laser pointer lens to make a microscope, and I'll show a video later. I think the best representation of what this instrument is trying to achieve is through this little magnetic stir. In the Dropseek protocol, they call for a very expensive piece of equipment. It's $1,000 to basically stir a disc in a tube to keep the microparticles in suspension. And here I've basically replaced that piece of equipment with a $2 magnet, a $5 motor, and a $10 motor controller. And so that achieves the same kind of thing compared to this other larger piece of equipment that's much more expensive. So it's much lower cost here. And the device can operate with a couple of number of inputs. So you can either have one input across multiple options for those inputs, or two, and ultimately three, with a small restriction on two of the inputs being the same pressure. So you can perform different types of droplet microfluidic experiments on the same instrument with very little changes to the instrument. Okay, so this is the traditional Dropseek chip, and here is the Dropseek chip that I had to modify for the MiniDrops instrument. It's very similar. However, you can see these are kind of 3 inputs that are nested. And in my case, for this instrument, I basically split these channels for the cells and the microparticles that are going into the droplets that I described earlier, and you collect the droplets from an outlet. You can see a nice video here in the corner. These dark circles flowing into this microfluidic chip are actually those barcoded microparticles I was telling you about. Here you can see the droplets being generated at the end. It's kind of a fuzzy line here because the camera is not very good at resolving the high generation rate of these droplets. But something to notice here is you can see the cells actually flowing into these droplets. So you can use a Raspberry Pi camera with a small, inexpensive lens, and you essentially have a microscope that typically would have cost at least $1,000 or more. And this instrument reliably generates droplets with monodispersed droplets with very, very precise diameters, and we can load these microparticles into the droplets in the Dropseek experiment, and it follows the standard nice Poisson distribution of loading. So we set out to basically benchmark the instrument, and what I'm showing here are 2 human mouse plots at different cell concentrations. So that this experiment allows us to basically measure the degree of doublets in our experiment, in our Dropseek experiment. So essentially, if the gene expression profile we recover from the Dropseek experiment is aligning to these, as we would expect a mouse, we plot it as blue up here. If it has human transcripts, we plot it over here. If it has genes from both, we can easily identify this as a doublet, shown as purple here. And what you can see is that these experiments show that we have a tunable doublet rate. So if we want more cells, we have to accept the fact that we're going to have more doublets. And this is actually proportional to the cell capture rate, which these are 2 different cell capture rates. In order to quantify the accuracy, we used a spike-in mix of ERCC molecules. And in order to look at the sensitivity, we can also look at the number of these ERCC molecules, essentially the UMIs per microparticle or cell barcode for each of performing Dropseek, and then essentially Dropseek on this MiniDrops instrument. And again, here's a bioanalyzer trace just showing that the profile of these well-known ERCC molecules is identical for both techniques. So really, we're not really changing anything here. This is really the Dropseek experiment, and it's just happening on the MiniDrops instrument, which is essentially identical. Okay, so at this point, we set out to use the MiniDrops instrument to apply it to a clinical study. And the disease we wanted to look at was rheumatoid arthritis. Rheumatoid arthritis is an autoimmune disease, and it affects approximately 1% of the population. While the cause of RA is not known, the etiology of the disease is hypothesized to originate from a combination of environmental and genetic factors. For example, if you're a smoker, that's a risk factor for RA. Also, women have a higher incidence of RA compared to men. Clinically, RA presents itself as multiple tender and swollen joints. In addition, there's autoantibody production, typically rheumatoid factor and ACPA, in addition to cartilage and bone erosion. So in RA, the synovial membrane lining, which you can see here indicated by the black arrow, is expanded to about 10 to 20 cells deep. And synovial fibroblasts take on this aggressive phenotype marked by the expression of disease-relevant cytokines and ECM remodeling factors. Okay, so this is kind of the breakdown of the experiment. We have RA patients, and they had come in and they would be undergoing a surgery, either synovectomy or arthroplasty. We immediately received the tissue, disaggregate it, and then run it on the MiniDrops on site. So the important thing about the MiniDrops is that it's very small size, so we were allowed to bring this to a lab adjacent to the clinical research area, and the patient would undergo surgery, and then we'd immediately bring the sample to the MiniDrops on site. Now, this is in contrast to experiments where you might, be performing surgery and then have to transport cells somewhere else, and this is kind of a more complicated process. So we brought the experiment to the location of surgery rather than the other way around. In all cases, the joint we were looking at was the knee. For this study, we looked at 5 different patients and performed a few replicates on some of those to assess the technical reproducibility of the instrument, but also to learn some biology. So Alex did a nice job of explaining the single-cell representations here, so I won't belabor it too much, but this is a T-SNE representation of the 20,000 cells that we were able to sequence from all of these patients. Every dot here is one cell, and we've colored them based on the cell expression, the gene expression profile for these across all patients. And we can see that we reproduce all of the main immune cell populations that we would expect, and we also have a large fibroblast population here as well. So in the T-SNE below here, I've colored the dots or the cells based on the patient that they came from. And you can see in each cluster, all colors are present, so it lets us know that all cell populations are represented across all patients. Also in this graph, we're looking at the distribution of the different cell types across patients as well. You can see there's some variability here. It's hard to tell where this is coming from, if this is actually due to the different surgical procedures that were occurring, or the particular surgeon, if got more tissue from one patient than another. However, we also performed replicate runs on an identical tissue sample from patients that had undergone surgery, and you can see the cell type fractions shown here are highly similar for these technical replicates. So we also looked at gene expression between identical cell types in different patients, and these were very highly correlated. And we also, as a sanity check, looked at gene expression across different cell types, and they're not as highly correlated. This is just a sanity check to make sure we were understanding what's going on. These are some feature plots that I want to go through very quickly. So we can display genes of interest and investigate how well these are represented across the different cell types in our clustering. In these images, purple is representative of higher gene expression, whereas gray is lower gene expression. And we kind of make all of these clusters light up based on this gene expression. So for example, CD3 here, which is a pan T cell marker, lights up the T cell cluster as expected. We can also look at CD4 and CD8 gene expression, and these are largely the genes that were probably making this clustering, and you can see CD8 and CD4 are in the CD4 and CD8 T cell populations as expected. Collagen 1A is present in the fibroblasts as expected, as a major component of the ECM. Marco, a gene for macrophages down here, is only in the macrophage cluster. Granulysin is in natural killer cells and cytotoxic T cells. So everything makes sense here. So within the CD4 positive T helper cell population I showed on the last graph, we detected very similar markers represented by MAF, CXCL13, and PD1, which had not previously been identified in single-cell RNA-seq studies of human PBMCs. However, there was a recent CyTOF analysis of RA synovial tissue, which identified this population with consistent markers that we had identified as well. And they called this a peripheral T helper subset. And they, in their study, determined that it would support B-cell activity and autoantibody production in this non-lymphoid tissue. So we also performed pathway analysis tailored to the single-cell data and identified functional modules. You can see down here is the pathway analysis. Functional modules upregulated specifically in these cells, including the regulation of inflammatory cytokine production and B-cell differentiation. So this kind of demonstrated our ability to identify cellular phenotypes that are relevant to the disease tissue in question using single-cell RNA sequencing in a clinical context. Okay, so we wanted to take a closer look at the fibroblasts. So our unbiased clustering returned 3 distinct populations, and one of them was sort of a subset of the other population. So we kind of annotated these as fibroblast 1 and fibroblast 2, whereas a 2A is a subset of the larger, 2A and 2B are subsets of the larger 2 group. So we look to validate this major separation in our fibroblast data by performing differential gene expression analysis. And we can see some of the markers that popped up between these 2 populations, 1 and 2 here, namely CD55, TIMP2, MMP3, I think Thi1 is over here as well. So we can take these features, put them back on the TSNE graph and see how these color our populations. And again, you can see CD55 and Thi1, which we were particularly interested in, show this nice split in these fibroblast populations. So this is something that we wanted to look into a little further. So we set out to validate this separation in the fibroblasts and basically test the specificity of these markers using complementary techniques. So first we use flow cytometry to demonstrate that CD90 antibodies and CD55 antibodies stained these independent populations, which they do, which you can see at the top. We also performed bulk RNA-seq on CD55 and CD90 flow sorted fibroblast populations, and the expression patterns are very similar between different patients and also agree with the single cell data. Finally, we wanted to perform immunofluorescence and see if these fibroblast populations were located in distinct portions of the synovium. So what we did is we stained for CD55 and CD90, and you can see for CD55, this is actually following the lining of the synovium. So this is a synovial fibroblast marker here for the lining specifically. And CD90, it was more interstitial staining with some brighter spots interspersed kind of in the interior of the synovial membrane. All right, so that kind of closes the clinical part of it. I just wanted to, at this point, just say we open sourced the MiniDrops. Everything is online on this open repository for essentially microfluidic systems called metafluidics. The circuit board design, the 3D printer files, the chips, microfluidic chip designs that we used, the code is up there. Everything's up there for people to build their own instruments and perform their own experiments. So at this point in the talk, I want to kind of switch gears and talk about another technology that was developed in our lab. And this was taken on by Marlon Stoikius. And so the question that he was asking was, can we somehow use antibodies to read out protein abundance in a sequencing experiment, a single cell sequencing experiment? So typically we're using antibodies with, conjugated to fluorophores to do immunofluorescence and all types of sorting, flow cytometry. And so the question is, how can this be adapted so that we can read this out on a sequencer? Namely, how can we get a sequenceable readout from an antibody? So Marlon's idea was to use an oligo tag that mimics a transcript, right? So adding an oligo to an antibody wasn't necessarily new, but making the antibody appear as an mRNA with a poly-A tail was kind of the major innovation here. And so the way that this works in a typical single-cell RNA-seq experiment is that you have an antibody that's conjugated to an oligo that has a handle, an antibody barcode that basically, once you read this out in the sequencer, tells you exactly what antibody this is. And the number of possible barcodes here is much larger than the number of available antibodies, so there's no issue with crosstalk in terms of antibody identity here. And finally, we have a poly-A tail to make this oligo conjugated to an antibody read out in a single-cell RNA-seq experiment. So this is sort of what the experiment would look like. First, we're going to stain cells with these antibody oligo conjugates in the same way we would stain them for flow cytometry or FACS. And then we wash the cells to remove unbound antibodies. And then we can directly port these into a single-cell RNA-seq droplet microfluidic device, such as 10x or Dropseek, what have you, and basically follow through the protocol. Now, the antibodies that were bound to these cells are now going to be encapsulated in the same droplet as the RNA derived from those cells. So both of these will get, attached to the microparticle via the poly-T, poly-A hybridization. And after breaking the emulsion, you can perform RT in bulk, typically as you do in a Dropseek experiment. Now, at this point, the antibody oligos are a much smaller size, and typically we size select for the larger things after RT in a Dropseek experiment. So in this case, we just keep the smaller stuff as well, which is the ADTs or the antibody-derived tags. And in this way, we have the RNA from single cells, and we also have information from the antibody tags. And they are identically barcoded, so we know for each single cell that which antibody bound to those cells. Okay, so to kind of demonstrate this technique with a variety of different antibodies, these experiments were performed with cord blood cells, and the cells were stained with a panel of 13 antibodies for well-known canonical markers. And so we ran the site-seq protocol on 10x and obtained data for about 8,000 cells. And so, again, what I'm showing here is clustering based on the RNA. So you can see the familiar immune cell populations showing up again. CD8 T cells, CD4 T cells, monocytes, natural killer cells. And so we can visualize gene expression in very similar way as I showed in the previous experiments. But in this case, we can also now identify proteins that were bound to these single cells, as shown in these green plots below each one of the corresponding markers. And one important point that I want to make is that for these single cell experiments, we're often dealing with a lot of dropout at the RNA level. And so down in these lower panels, you can see the mRNA for example, CD11C, very sparse at the RNA level. And with the antibody readout, the ADTs, we can see is much more highly representative in this cluster. Okay. So we also wanted to determine the dynamic range of CiteSeq. We call this technique CiteSeq. It's cellular indexing of transcriptomes and epitopes by sequencing. So we wanted to directly compare it to flow cytometry. So in this experiment, we got CBMCs, stained them with antibodies that are conjugated to fluorophores, and also antibodies that are conjugated to these oligos. These cells were actually sorted into 4 different gates depending on their CD8 expression, very high, medium, low, and lower, I guess. And then we performed flow cytometry on these, and in addition to CiteSeq. When we analyze these, we can see CD8 levels that are very concordant with the flow cytometry data. Here's showing the CiteSeq readout and flow cytometry. You can see the populations that were sorted are very similar in their counts between the 2 techniques, showing that we have very comparable relative intensities in CiteSeq as we do in the flow cytometer. Okay, so we've also expanded this technique to even more antibodies. So we're typically doing experiments with on the order of 100 antibodies that can be stained onto these cells. So in this case, I just wanted to highlight the kind of application of doing something like this. Basically, this is T and NK cells only, and what we see is much higher resolution and larger number of clusters that we're reliably able to identify using this technique that we wouldn't otherwise using mRNA alone. All right, and finally I want to talk about an extension of CiteSeq that we call cell hashing. And so essentially what we're going to do in this technique is barcode similar antibodies, identical antibodies with different antibody barcodes. So in this case, there's an identical antibody here, but these oligos have the same structure as I showed previously. However, they have different barcodes. And what we can do with this is if this antibody is for a ubiquitous cell marker, we can now stain our cells, split our cells, and stain them independently to perform sample multiplexing on a device such as the 10X or Dropseek. And so that's exactly what I'm showing here. We have 8 genetically distinct donors, and we split them into pools, stained them independently, and then repooled them, and then flowed these through a standard droplet microfluidic workflow, in this case 10X. You can perform the standard analysis and then demultiplex the data downstream to identify the cells from each patient, each donor, or each sample that you've used over here. Okay, so first you can simply analyze the RNA data and observe the cell types that you would expect from blood. Again, we're seeing all the same cell types. They're colored here by the different sample separation. So you can take this information, the hashtag information from these experiments, and now we can demultiplex it. So what does that look like? There we go. So this is now clustering that we're basing on the cell hashing. So we see 8 very large clusters, which are the 8 samples that we split into these different pools and stained independently with hashing oligos. And we also see 26 smaller clusters that are all of the possible combinations between the hashtag oligos, and these are essentially the doublets that kind of pop up in a normal single-cell RNA-seq droplet-based experiment. And there's exactly 26 of these indicating that you have all possible combinations of doublets between these different barcodes, these different samples. So we can easily sort of throw those to the side now and with relative confidence say that these are doublets and we don't want to analyze them. So this is what I'm showing here is a heat map now of the demultiplexed signal. And you can see it's highly enriched for the expected number of cells. So in this experiment, we get, this technique also allows us to overload the system in a way. So we get 25,000 cells in a standard experiment now. And we can easily say that 20,000 of them were independent single cells. You can see here, these cells are all coming from one barcode, these are from another, and so forth for all the different hashtag oligo signals. And then we also recover all of the possible multiplet signals here as well, and a negative control which shouldn't have stained for any of these antibodies. So the end result here is that we can reliably load multiple distinct samples into a single cartridge for single-cell RNA sequencing. We can process them together and then computationally deconvolve them downstream after sequencing. So we can save money and we can also increase throughput on these experiments at the same time. All right, so just to summarize, I talked about MiniDrops, which is a low-cost open-source instrument for droplet microfluidics. We demonstrated the Dropseek protocol on the MiniDrops in a clinical setting to identify novel subsets of synovial fibroblasts and confirm a T peripheral helper population that had also been identified in CyTOF experiments. I also told you a little bit about CytSeq, which allows us to detect cell surface markers and the transcriptome of single cells. Because the libraries resulting from the antibody signal are less complex, there's much lower dropout compared to RNA. The readout is comparable to flow cytometry, but in this case we're allowed to get many more markers because we're not limited by fluorophore overlap such as you would be in flow cytometry. We can have as many barcodes as we want, essentially. And we also have the added benefit of now getting the transcriptome as well from each single cell. We think that this technique is basically compatible with all oligo-DT-based single-cell RNA-seq platforms. We've tested it obviously on Dropseek, 10x Genomics, and also we tested DDSeq. And I also showed you briefly that we can increase the resolution of the cell types and states that we can discover using this technique, using the extra protein information that we get from this technique. And finally, I told you very briefly about cell hashing, which is a sample multiplexing experiment. And we're allowed to superload these instruments to obtain up to 25,000 cells in a single run, and then basically throw out the doublets because we can now identify them more reliably. So with that, this is probably the most important slide on the whole presentation, but a lot of people worked on a lot of these different techniques, but I really want to thank Rahul, who was integral in all of the techniques that I told you about today, Andrew and Laura at the Hospital for Special Surgery, Andrew at NYU and the Genome Center for doing a lot of the analysis, and also Marlon, who really championed the CytSeq technique, and Peter, who's our group manager, who's been very integral in everything we do, including the CytSeq and the cell hashing experiments. All of the protocols for CytSeq are online. You can check out this website and follow us on Twitter, and thanks. I'll try to answer any questions.
Thank you so much, William, for your perfect talk. Can I ask the first question? Sorry. How many samples can you multiplex using approach of hash?
Yeah, so we're not sure. I mean, you can make as many barcodes as you want. I guess it really depends on your patients and splitting, one sample into that many or dealing with that many samples. But I don't see a major technical limitation as to the number of samples that you can multiplex. You're mostly limited by, for the most part, we do 10x. So if you multiplex in this manner, you can get up to probably 25,000 cells, of which 20,000 will be single cells. So I'd imagine at some point, if you split your sample into too many samples, or you have a lot of samples, you're going to have less cells per sample, right?
Sure. Yeah. Thank you.
Thank you, William. Wonderful presentation. Just a few years ago, I started with a single cell analysis, and I started with a Dropseek. And one of the main problems that we found is in every drop, it was really difficult to load one cell. Generally, it was loading 2 or 3 cells per drop. How you solved that with your system?
So in terms of the MiniDrops and Dropseek, yes, so every drop is not going to have a single cell. And in fact, most of the drops that you generate in that experiment are not going to have anything, not even a single cell or a bead. About 10% of the time you get a single cell, about 10% of the time you get a single microparticle. And you're looking for the overlap of those when they occur together into a droplet. So in reality, the number of droplets that you're making is very large. And so the number of droplets with a cell and with a microparticle, which is what you're looking for, is much smaller than that. So it's a small fraction of the overall. So you shouldn't expect that every droplet should have a cell or a bead. And if you do encounter 2 cells within a droplet, that happens. It's statistically you'll get it according to the Poisson distribution, but it could also indicate that protocols that you use may not be fully dissociating the cells, or there might be other issues at hand there.
Thank you.
Yep.
Yeah, and while I'm walking, just a question. Is there any way to do it not only for the membrane proteins, I mean CytSeq, but also for intracellular proteins?
Yeah, so whenever we talk about CytSeq, this is the question that everyone asks. Can you get this to work for intracellular, right? Everybody's interested in that. I know there's a lot of people working on it. We've heard some things that some people have it working. We tried extensively, and it's very difficult to optimize the conditions to essentially fix a cell and then stain it intracellularly, and then also retain the RNA from that fixed cell. That's the tricky part, right? To get good RNA from a cell that's been fixed in these experiments. So it's difficult. We're working on it, but we also know that a lot of other people are working on it, and actually we expect something probably very soon along those lines. Yeah.
I have a question. Is it possible with MiniDrops to isolate cells and for the same cell to do both methylation and single cell RNA-seq to know how they interplay?
Right, so that it wouldn't necessarily be, the question isn't necessarily if MiniDrops can do that. The question is if you can do that with droplet microfluidics at all. And I'm not entirely familiar with if there's an approach out there that can do both in droplet microfluidics, methylation and RNA. There might be, depending on how complex the microfluidic chip is and the requirements for that chip, it may or may not be able to be done on the MiniDrops. I guess the answer is I'm not sure.
Okay, thank you. And could you give just examples for kind of a price tag? I mean, if you have some, I don't know, 5,000 cells with some amount of million reads per cell, how much it can cost to the lab?
Yeah, I don't have a precise cost for you. I don't know that number offhand, but I can tell you that generally in these single cell RNA experiments, I think we sequence to around 50,000 reads per cell. So just multiply that by the number of cells you have, and then do the conversion for how much sequencing you need, and that'll generally give you your cost. But we typically do 50,000 reads per cell.
Questions?
And yeah, hey, so did you try to compare the results of your MiniDrops with like other microfluidic technology?
So we only did those basic experiments where we compared it to Dropseek on the standard setup, and they were, I mean, it's essentially identical, as one would probably expect. I'm not changing the Dropseek protocol. I'm just doing it on a different instrument. But there may be other droplet microfluidic protocols that would be compatible with MiniDrops. I haven't tested those in the two different settings. So.
Fantastic talk. So, do you know of anyone trying to combine the MiniDrop with long-read sequencing, like nanopore?
No, I don't. How would you? For what purpose? I don't.
Yeah, to see the variability in the isoforms between the individual cells, because that's something interesting.
I see. No, I haven't. Yeah, haven't thought about that approach.
More questions? Yep.
All right.
Finished? Okay. Thank you so much. It was very exciting.